Okay, so now it's my pleasure then to welcome the second speaker of today, so Professor Caitríona Lally. And well, Caitríona is a professor at Trinity College Dublin and professor in biomedical engineering, and then she's focusing on arterial material modeling, so including tissue modeling and also problems that are related with the remodeling of the collagen. So in order to predict possible problems after, for example, stenting, atherosclerosis, plaque instability. And she has been recently combining a lot of modeling with experiments. I don't know if you will talk about experiments in the end. Mm-hmm. So which is following the same line as a previous talk of Philippe, but with a different system. So I actually also came to know Caitríona when I was at the very beginning of my PhD at a congress with Patrick Prendergast.
Previous supervisor.
And she had a talk, and so she was about halfway or about to finish her PhD, and then she had a talk, and she was very frightened because she only had a simple material model for a neo-Hookean model. So those who are doing osteomuscular modeling with me know what I'm talking about. So one parameter model. And
I don't think I should.
and she was very worrying about this because she was saying, oh, no, that's over simple. They will kill me. Yeah. And and then so in the end, so Caitríona received progressed, and she received a lot of awards, including an ERC recently. So actually,
Yes, you can start.
Going simple hasn't been that bad.
Yeah, keeping it simple can be tough, actually.
Yeah. Okay. So.
Yeah. Thank you, Jerome. Thank you very much, and thank you for the invitation to be here. You can imagine I'm from Dublin, so if anybody's been to Ireland or probably ever heard about it, you know it rains a lot. So of course, when I got an invitation to come to Barcelona, I thought, fantastic, Barcelona in the summer. And then I arrived yesterday, leaving the sun in Dublin, believe it or not, behind me. And as we got here, it was streaming down. So I'm delighted to see today is better. So that my visit has not been without some sun. So that's great. So actually, Jerome says Caitríona, but I'm known as Trina. So again, for maybe the social events after, that's what I'll respond to. So if anybody wants to talk to me, I'm very happy to chat to you later. So because I'll probably, like a lot of lecturers, I talk too much. So I'll probably end up using the full hour talking now. But I really would welcome discussion and so on. So I'll be around for the rest of the day today. So very happy to answer questions. So what I want to show you today, I know again this is about in silico modeling and so on, but what I want to show you, I suppose, is somewhat, in the same kind of vein as what Philippe showed earlier, is this idea of using both in silico and in vitro testing. In some cases to understand, so we'll use the in silico model to actually understand mechanistic responses of tissue, and in some cases as well for clinical applications. So hopefully I'll get the time to show you all that. So as I said, within this talk. So all of my work, unlike the earlier talk, is in the cardiovascular area. I have focused on this area my whole career, so I'm looking at arterial biomechanics mostly, and now some valve biomechanics as well. So this is because, basically, as most of you will know, because cardiovascular disease is a leading cause of death. And in fact, in Western developed countries, it's been maintained the leading cause of death for some time. And in fact, nearly half of all Europeans, I'm afraid, will suffer death because of cardiovascular related disease. So this usually manifests itself in a couple of ways. So this is through either arteries blocking, through atherosclerosis. So this is where the arteries, fatty deposits are laid down, and we see changes in the underlying structure of the artery, and the artery can ultimately totally occlude or be occluded so much that it will have effect on your health, okay, and the oxygen being delivered to your tissues. The other area that we see cardiovascular diseases manifest themselves is in aneurysms, which is quite a different a mechanism than what we see in atherosclerosis, but again can be fatal. So in this case, what you're talking about is actually the underlying constituents of the tissue can degrade and alter, and that can result in the tissue actually starting to expand because it doesn't have the appropriate load-bearing components within it. So this would be particularly collagen fibers within it, and there will be elastin degradation too. But our most significant load-bearing component in our arterial tissues are the collagen fibres within that tissue. So when these degrade, what can happen is that the artery will start to expand uncontrollably, and if left untreated, which in many cases we're not even aware an aneurysm is progressing, it can result in a ruptured vessel. And this can be fatal, particularly if it's some of these larger vessels, like the abdominal aortic aneurysm, in the abdominal aorta, I should say. So here. Probably even just to mention at this point is why we're so interested in arterial diseases and understanding arterial structure. There are a lot of interesting facts about where these types of diseases progress. So atherosclerosis happens predominantly in areas like coronary arteries. You all maybe have heard of somebody having blocked coronary arteries and it results in a heart attack. And in that case, these are arteries that are predisposed, can actually have this atherosclerotic development. But it's interesting to note that there are vessels, so femoral arteries are also predisposed to atherosclerosis. Carotid, which I will talk about in some of this work. But it's worth noting that some vessels also seem to be spared. They don't seem to have the same risk for the same individual, even with their same lifestyle and so on. So that's something interesting for those of you out there starting out, maybe if you're doing your PhDs or you're thinking about research questions. That's one that really hasn't been answered yet, as to why is the radial artery, for example, preserved when it comes to atherosclerosis, even for individuals who have sedentary lifestyles or poor lifestyle conditions like smoking and so on. And the same can be said for abdominal or aortic or any other aneurysm. This degradation process, it happens is more likely to happen in certain vessels than others. So it can happen particularly down in this abdominal aorta, down into the iliac arteries, into the legs. Happens again, you can see some of it down into those vessels in the legs, but again, the other area that you'll see it commonly occur is in cerebral arteries in the brain. And again, we do look at some of those vessels as well in our lab. So again, don't see aneurysms happen as much anywhere else. So again, radial artery doesn't occur. So just interesting to note that. So some background on arteries, as I've mentioned, they actually have a number of different components. I mean, there's elastin, there are a lot of gags present, smooth muscle cells. There are other cells like fibroblasts and so on in the outer adventitia. But the thing that I'm really going to focus on which I focus on in my research and also within this talk, is really on the collagen fibers. And this is because, like many other tissues in the body, the collagen fibers in arteries are the ones that really bear the load. And they have a complex arrangement within arterial tissue. So I made it look very simple here in that these are kind of a hatched crossover fiber pattern. But a lot of people, this will represent it like this, which are these kind of helical fiber patterns that go around predominantly in the circumferential direction, but not exactly, okay? So we'll see that as we look at remodeling later as well, how that angle is dictated. So it is collagen fibers within this arterial structure that give rise really to the strength of the artery. So in things like rupture, you won't see these collagen fibers in their optimum, both in the quantity and in the orientation that they need to be. And the same, you'll see that it's remodeling of these collagen fibers that might dictate then the health of this tissue in diseased tissue, such as, in atherosclerosis. So it's this maladaptive remodeling. That's just over there, a polarized light microscopy image of these collagen fibers. That's the adventitia actually there, here taken in our lab. And you'll see, they're quite wavy in the adventitia. They're more aligned as we move in towards the inner layers, into the media and intima of the vessel. And I'll show you some of those images later. But it's really maladaptive remodeling. So it's this where the remodeling is not optimized that we have problems in this tissue, okay? Where we'll see there may be issues with rupture, for example. So I'm going to just talk about a few areas where this remodeling of collagen is key. So I don't know if anybody has any idea what that is. Anybody know what that image would be? So that is tissue that has been harvested out of the body. It's actually a plaque, okay? So it's at a bifurcation. So it's here in the carotid, in the neck, okay? So there's a surgical procedure that can be carried out where someone has occlusive atherosclerosis, so atherosclerosis blocking the vessel. You can have, it's not, something we'd all want to sign up for, I think, but you can have your neck cut down here, opened out, and your artery, your carotid artery, opened and kind of the plaque delaminated out of that vessel and removed, okay? So this is done for individuals who have this occlusive symptoms related to this carotid plaque being present in their artery. And these tissues, when they are removed, we see they have an interesting fiber structure, okay? So this is of interest to us. We can, and I'll show you later, we image some of these vessels before this procedure called a carotid endarterectomy is carried out. And then we obtain the plaques after the procedure, and we're able to look at this mechanically. So why would remodeling or fibres within this plaque be relevant? And this is because these plaques, some are very at risk of rupturing and some are not. Some of these plaques can progress over time. They can block a vessel, but they can actually be quite stable. And the vessel and its collaterals can actually develop to accommodate the blood. And so we've no symptoms related to it. And in fact, rather than having this very invasive surgical procedure, you're probably better to leave that plaque. It's safer to leave it, provided it's stable, provided it's not at risk of rupturing. Obviously, if it's at risk of rupturing, what can happen is bits of that plaque could break off and they can flow upstream with the blood and end up occluding a small vessel in the brain, such as the cerebral arteries, and then you will suffer a stroke. So at the moment, there really isn't a way to say whether a plaque that is present when we image it in a particular vessel, we can see how much it's blocking the vessel, but we actually don't know if that plaque is likely to rupture or if it's not. And at the moment, mostly what we do is we just, the clinicians should say, what they do is they'll treat a patient where there's basically occlusion of the vessel greater than about 70%. And it depends on the clinician. They may carry out an intervention earlier than that. But in some cases, that's not necessary, okay? So what I'm going to show you today is some of the modeling that we do, but also I'll mention that it is understanding this structure. So maybe you can turn on that. Should play, there we go. Maybe being able to see what the structure of that plaque is, not just how much it's blocking a vessel, but underlying structure. I'll show you that later on because perhaps many of you are not aware of the techniques that can be used to see this. So remodeling and understanding the fibre structure here, we believe is key to understanding the stability of that plaque because collagen fibres are the load-bearing component. If they're aligned in the right way, then they can give that stability under the blood pressure. Another area where remodeling is key is in interventions. So in some cases, carotid stenting can be carried out to open up the vessel. You're still a bit at risk of some of that plaque breaking off, but it can open it up and restore blood flow. But in the cases of once we load that vessel in any scenario, so whether that's in the carotid down here, or whether it's more generally where we see millions of stents going in worldwide every year in areas like the coronary arteries, what we'll see is the loading that places on the vessel can actually induce remodeling. Okay, so remodeling and changes in the structure and the content of a vessel is all driven by loads, by mechanics. So these type of interventions are going to change the mechanics of that vessel and will induce remodeling. And we want to better understand what level of remodeling. Is there a critical load level that the remodeling could be negative for the vessel and its strength, or for consequent biological mechanisms. So things like when a stent goes in, we can also see growth of tissue inside those stents, such as in-stent restenosis, which can limit the success of that procedure. And again, this is often driven by this remodeling process. The other area that we're interested in terms of remodeling is bypass. So in cases where we can't carry out in this minimally invasive way, this approach of opening up vessels using stents, what will happen is these blockages will have to be bypassed using the likes of a bypass graft. Some of these bypass grafts are synthetic materials, and so they won't remodel themselves. But the areas around where the grafts are attached to the vessels, we see a lot of remodeling. And this can be driven by things like the mismatch in the stiffness of the graft versus the native artery. And we can also see tissue-engineered vessels being used in this application. And again, understanding the loading on those vessels through this attachment will dictate the remodeling in this tissue. Other areas, I've mentioned tissue engineering there, but tissue engineering not just in arteries, but obviously beyond that in other soft tissues. So in valves is key. What is the loading? How will that induce collagen to be laid down or collagen to be removed? Or other components as well. But as I said, particularly interested in collagen being the load-bearing component because it will allow this valve to function ultimately versus perhaps not functioning under the high pressures that we see in areas like the mitral valve and the aortic valve. And finally, I just mentioned, because I do work in this area as well, that there are many bioprosthetic heart valves out there as well that have tissue, the leaflets of those valves made from biological tissues. Some of that tissue most commonly being things like pericardium, the sac that surrounds the heart from animals, so bovine or porcine pericardium. And actually the realignment of fibres there can also play a role. Even though that's not living tissue, the actual realignment, the change in fibre orientations because of the load on the tissue can dictate the longevity of many of these valves. So these are all areas where I think it's critical that we understand how collagen alters its alignment. So if we think about each of these, so some of these areas, this collagen remodeling, I'm going to show you kind of 2 main case studies and discuss a few future directions, hopefully within this talk, okay? So the first one is really to focus on arterial remodeling using in vitro and in silico methods to quantify and explore particular aspects of collagen remodeling, which I'll discuss now, in arterial tissue. And then I'll look a little bit on damage, which overlaps and kind of continues a little bit from what Philippe showed earlier on damage models, and some future directions which are around this whole area of can we actually see this structure in vivo potentially in the long term? Can we see some of this relevant mechanical structure, the collagen structure? So the first study I'm going to present to you is work predominantly carried out by Dr. Robert Gall, who's a recent PhD graduate from my group, and it was a collaboration with TUE, with the Technical University in Eindhoven. So thinking again about collagen remodeling, remodeling is quite a complex mechanical driven process. It's not one of these aspects, it's all of these. It is the collagen fibers changing orientation. So this is just simply realigning because of the loads on them. It is collagen production, so we can have new collagen laid down because of the way the tissue is loaded. And we can also have degradation, that is collagen being removed because of, again, the loading being placed on the tissue. And in fact, what we see is that degradation, production, reorientation, they are all strain-driven processes. And degradation generally dictates production. So in the normal healthy tissue, what we'll see is just like with bone or any of the tissues of the body where we see this dynamic response to loading and so on, what we'll find is that degradation and production should balance each other. As collagen is removed, we'll have new collagen produced, maybe removal of damaged collagen, and we'll have new collagen laid down. When this goes wrong is when we see maybe this maladaptive remodeling. But also we'll see this reorientation as well. And the orientation of the collagen is key for bearing the load. So for this section, I'm going to focus a lot on the degradation. This degradation of the collagen and how strain drives degradation and also reorientation. So before we could do this, of course, we had to understand that we could visualize this collagen structure, this underlying structure. And there are many techniques out there for doing this. But most of them, as you'd expect, are either, they're all very time consuming, but they have limited depth of view, and in general, they're destructive. So we couldn't actually see then how that collagen structure was changing under load. We'd have to do many different tests over different tissues. And of course, there's variability in tissue. So we wanted to really see it in a tissue itself. So a technique that I'll show you shortly called small angle light scattering, which has been used for looking at non-destructively at the fiber patterns of valve tissue, valve leaflets, particularly bovine pericardial valve leaflets, and it was developed originally for that, but was also then applied to corneal tissue to look at degradation of collagen as the tissue was under load. So to look at it almost like in real time. We said we'll adapt this and we use it for arterial tissue as well. So we developed this technique for arterial tissue, but before we did this, we wanted to understand, well, what do we expect collagen to do under load in arterial tissue? What do we expect the degradation to look like? And when we looked at the literature, there were actually conflicting reports in the literature as to how collagen might be influenced by strain in terms of its degradation, which would then subsequently stimulate production, for example. So in terms of, if you look at a few of the different studies, Wyatt et al. Had identified that actually, as you increase the strain on collagen, what you would see is that the degradation rate would go down. So they were saying increased strain, you'll see reduced degradation. But actually another study then showed that actually in bovine pericardium that you'd see an increase in degradation as you increased the strain, as you can see there. And then there was a study on reconstituted collagen and almost pure collagen that had identified that there is a strain-dependent mechanism, but it is very much the strain level dependent. And it kind of brings those two studies, those two conflicting studies, and explains them in that what it identifies is that at low, now if you can see this, no, but at the lowest levels, no, it's not coming up there, is it? Sorry, so at the lowest levels of strain, what you see is that degradation is increased. As you increase the strain level now to kind of within a level that we would see physiologically, what we see is that degradation reduce. And as you go further up now into more pathological strains, maybe strains that we shouldn't see unless something has degraded or we're putting in a medical device, we see again further increases in degradation. So this again is not surprising in some ways. If you look at bone, for example, it'll behave somewhat like this. If we don't use it enough, it'll resorb. If we use it too much, it may potentially become permanently damaged and have accumulation of fracture of cracks, which will ultimately drive towards microcracks, which might ultimately drive towards fracture. So this was not unique to this tissue. So this was at the pure collagen level. So what about then at arterial tissue? Well, arterial tissue has a lot of collagen, but it is not pure collagen. So what we wanted to see, will this U-shape degradation curve or behavior be evident in arterial tissue when it's got all these other constituents as well? So we looked at the structural changes in terms of how collagen degrades in arterial tissue, specifically the collagen, then mechanically the tissue itself, and then you will see when I show you the data why we had to go numerically, because we couldn't fully understand the behavior that we saw. And this is why I want you to see this idea of this combined efforts of in vitro and in silico is really helpful for understanding the mechanisms of, for example, in this case, degradation in a tissue. So structurally, as I mentioned, we developed a technique, small angle light scattering system. So this is a very nice image provided by my PhD student who shows the helium neon laser, focusing lenses, and the sample positioner for the sample itself, and a camera then that looks at a projection plate as that laser light raster scans through the tissue. So each time the laser is placed through the tissue, that's an interrogation region, and you get this nice ellipse from the laser light, and then you get these multiple interrogation regions. Oh, you have one. Okay, thank you. Thanks. So here you can see that laser light passing through the tissue, and now I have to work with two of these at the same time, but okay, so here we can see this light here. This is what the backscatter will look like, and it's basically an ellipse which is actually refracting or scattering the light, I should say, from that laser beam that's passed through the tissue. And predominantly, it scatters the light at 90 degrees to the fibers which are acting like a diffraction plate as the light passes through. And so from that, you can find in each of these interrogation regions the actual dominant fiber direction, and you get a measure of alignment as well across the tissue. So this is, how aligned are those collagen fibers in the tissue, as well as what's the dominant angle of that collagen fiber. So using this, we were able to say, well, structurally, what does the tissue look like? So the intima, media, and adventitia, we took out these 3 individual layers, but we didn't have to section them as you do with histology or as you would with something like multiphoton, where you have to, you can only get a limited depth, or else you have to section the tissue to get further depths. So we were able to see for the intima, media, and adventitia, able to see that the fiber pattern in the intima was predominantly in the circumferential direction, as you can see there. I'll try and use 2 hands for this. So here we go. Yeah, see here. Whereas with the media, we can see again predominantly in the circumferential direction. That's what's represented here in this rose plot. And we can see, and then when we went to the adventitia, what we could see is that we had actually 2 fiber families. So in this porcine carotid tissue, what we were able to see is that the small angle light scattering could relatively non-destructively, as in each intact layer, see this fiber structure within the tissue. And so once we were able to do that, we could see how strain would control or alter that tissue and reorientate the fibers. So at 0 strain, you can see this is our cells plot here. This is our small angle light scattering, showing us the lines there representing the dominant direction, and the color map here representing the degree of alignment. So essentially, how dispersed are the fibers within each of those interrogation regions? Okay, so are they all in that direction, or are they more spread around that direction? Okay, what we can see here is as we move to 20%, apply a 20% strain to that tissue, we can see that the fibers now become very aligned with the strain direction, and we can see that both in the alignment. So now all the fibers in those interrogation regions are all in the same direction. And you can just note again here, just showing you that we actually used pins to load this tissue, and we were able to even see the reorientation of collagen fibers around those pins, and that's what's shown there in the last image. Okay, so just that the small angle light scattering could see this. So why was this useful? Well, now we could see the structure. We could see how it reorientates under load. We could now see, does that change as we degrade the tissue? So as we expose it to a degradation enzyme, we could see how that fiber pattern would change. So we took these common carotid arteries, porcine common carotid, we opened them out, made some sections for loading, first of all, to really see, can we see under different load conditions how this collagen structure alters? So do we see this U-shaped curve in this increased degradation at low strains, preserving the collagen at a kind of physiological strain level, and increased degradation at higher strains? So we looked at these 3 different strain levels, 0, 5, and 25%. Just, I suppose, worth noting why we looked at these particular strain levels. So I'll just show you over here on the right-hand side. If you look at the strains in a vessel, okay, the strains vary around this 5 to 8% physiologically in a healthy vessel. But when we do things like stenting, we strain that vessel way up, okay? We go up into that 20, 30% strain region. So we wanted to see, is that an area where degradation could be increased? And would there be kind of a critical level? And, in areas then, if you look at that kind of low strain level, once you have this atherosclerotic development within an artery, the artery will typically stiffen up. And now the collagen fibres in that tissue may not be experiencing that higher physiological strain level. They may be experiencing quite a low strain level in that stiffened vessel. So that's why we looked at these three. So what we did then is we put this tissue into a bath at 37 degrees C and placed bacterial collagenase within that bath to look at how this collagen would degrade over time in the bath. And used small angle light scattering to view the tissue all the time to see how that collagen fibres were changing because of that collagenase. So here's the control, and just showing you some schematics. There's our strain applied to our samples. And this is what, just taking a representative small angle light scattering, one interrogation region, which is like 250 micron by 250 micron. Looking at one of these, this is what the typical small angle light scattering images look. This is representative, as I said, across all of that sample. So what you can see here is that in the control, we have a particular fibre direction. As you keep it at 0%, that either stays close to the same or doesn't become any more aligned, maybe a little bit more dispersed, the fibres. But what we can clearly see is in 5%, our fibres become more aligned, which indicates that all the other fibres that are not being loaded at this level are starting to degrade more preferentially. And at 25%, what we see is that now our fibres, even being loaded, are degrading more preferentially because we're getting even a more dispersed result. Okay? So what does this mean? This means that essentially from our control through to our 0, 5% and 25%, it's saying that at a certain strain level, we're protecting our fibres. This is around the 5%. And either side of that, we are not. Okay, we are actually degrading the fibres that are being loaded and ending up with something that's not very well aligned in terms of its structure. We confirmed this by looking at histology, which again is a more standard approach to looking at collagen fibre structures. So this is polarized light microscopy images. And you should be able to see across those three, and here is the analysis of that the center images is more aligned. Okay, we can see that the fibres, whereas at either side of that, we have fibres in every direction. We're seeing a lot of dispersion. And we also confirmed this again with second harmonic generation imaging, so our multiphoton imaging. And we looked again there, we can see that at 5%, we're seeing a more aligned structure. And again, these are representative images of across that whole tissue. So just summarizing what we see there is actually, we can summarize this by looking at this change in alignment, or as we refer to eccentricity of those fibres across the tissue, and taking a raster scanning across the whole sample, we can see that consistently what we do see here is that under the 5% strain, our sample is becoming more aligned. And either side of that, it's either being kind of maintained in less alignment, or it's actually becoming even less aligned. Okay, so it's degrading now in the direction of load. And this indicates that either side of this more physiological level, this 5%, our collagen fibres are degrading because of the load being placed on them. So that has impact in terms of the structure and understanding our tissue structure and how it would be affected by load and collagen. So this is our representative image. But while this represents our U-shaped curve, so we do see what Huang saw with the reconstituted collagen when we degrade our arterial tissue, we wanted to see if structurally this is also happening. So when we take a full piece of tissue and we load it now with this bacterial collagenase present, do we see this behavior as well? So that was just interrogating the collagen, remember, because that was using small angle light scattering. Now we're taking the whole tissue. So taking that tissue and doing stress relaxation experiments, placed them in baths, looked at relaxation. You'll see here, there's the stress relaxation, but after that, if you were to allow it to remain in the bath without any collagenase present, the load will just completely level off, as you see there in the control curve here. Whereas what we did was there were some controls, but obviously we also placed samples into those baths and exposed them to bacterial collagenase and to see how does that, again, affect the tissue degradation. And we did this to establish a degradation constant for the tissue under different loads. So we did this for two different types of collagenase. One, the bacterial collagenase we'd looked at previously. This is kind of a crude form. It's used again, many people do use this as a representative degrading enzyme for collagen, but it actually isn't as specific to collagen as you might like. It actually does remove some of the matrix as well. But there's a purified collagenase that you can obtain that really only targets collagen. And you can see that there are 2 histological images that confirm that. Here you can see in the crude, you're removing collagen, but you're also starting to affect the overall structure, having some effect on the matrix. Whereas here you can see that's the collagen here is degraded on the outside there, and you've still some collagen remaining in the center. So you can see it's very selective for the collagen itself. So using this approach and these relaxation experiments, we were able to see that the degradation rate in the crude collagenase, this is what we obtained in circumferential and in the axial direction. So we got a linear response in the tissue. So this is now the response in arterial tissue. What's our degradation of arterial tissue when we expose it to bacterial collagenase and expose it to different loads, different strains? So here you can see the strain level that we investigated. So this isn't very like what we saw when we were just looking at the collagen itself. So this is the overall structure of the tissue. And it's linear. And why it's different for axial than circumferential, I mean, we'd expect this because there's an awful lot more fibers in the circumferential direction than there are in the axial. So it's not surprising that circumferential direction we see higher degradation than in the axial. But the fact that it was linear perplexed us a little bit. It's not what we expected. We expected to see this U-shaped curve. So then we said, okay, maybe it's because of the type of collagenase we're using. So we'll use the purified collagenase. We know that only targets collagen. And we look at the response. And then this is what we got. So we got a very, and again, this is a lot of experiments there carried out. We saw this increase, initial increase in degradation, then a marked reduction in degradation, and then a further increase again. So this, again, wasn't anything like our U-shaped curve. So this was not like what was seen for reconstituted collagen or what we'd seen when we were just using an interrogation method to look at the collagen itself. So this is where I would say your numerical modeling can come in. So this was, we had real experimental data. We knew this is what actually happens to the arterial tissue, but we didn't fully understand why. So we used a numerical model, which is to try and explain what we saw. And like Philippe earlier, we said we'd take a kind of more mechanical analogy and see, can we look at this tissue, what might be affecting this degradation? And because arterial tissue is that bit more complex than even, reconstituted collagen, pure collagen, the arrangement of collagen in this arterial tissue is quite complex. So if you take a strip of tissue, it has matrix there, but that matrix, which can be represented in our model by just having a constant stiffness, Km, these collagen fibers are embedded in this matrix, and they actually are crimped a lot of the time in this matrix. And the degree to which they're crimped will dictate how much load the collagen itself actually sees. So we created this model which could have the fiber stretch built into it. So there, this is a lambda F, there is our fiber stretch. And we then have this lambda P, which represents how much, basically, whether the fibers are in a crimped state or in a pre-stretched state. So altering this value for lambda P to being greater than 1 or less than 1 would mean that now our fiber stretch is either below 1 or above 1. So it's either crimped or it's stretched. So doing this, we were able to apply stiffnesses to these, and essentially you can just see that basically if it's crimped, our switch there is, our lambda F is below 1. We can straighten those fibers by using that switch between the values of lambda P and lambda F, and we can actually make lambda P below 1, and therefore our lambda F, our fibers are in a stretched state. Okay? So we use this 1D model to look at this degradation response, where we now modeled the collagen purely by this U-shaped curve, where we know it's got this collagen at low degradation, it'll be protected at 5%, or I should say at low strains, it will be preferentially degraded. At a kind of medium level strain, it will be protected, and at higher, it'll be further degraded. And we used our crude bacterial collagenase to model this matrix reduction because we knew that it was being also degraded with the collagen. So we then considered, well, what is the arrangement of this tissue? We don't have the same degree of collagen crimp throughout our tissue. We see this actually in histology. You can see here, this is a histological image that shows you the degree of crimp in the tissue from the inner to the outer in an unloaded vessel. Now I stress that because in a loaded vessel, we typically see that this crimp becomes quite uniform throughout the vessel because again, it's this kind of homeostasis within our body. So what we see that if we took strips of this tissue, so we took that vessel and we opened it out, and that exaggerated the degree of crimp gradient across the vessel. So we hypothesized that it was actually this degree of crimp change that might be driving that result that we got of the degradation. So here we can see we created now our fiber matrix unit. We put it in parallel as multiple units, an array of units, and we're able to then represent the degree of the change in crimp across the vessel. And we used this model to represent then the stress in the tissue, the overall stress being a combination of the matrix stress and the fibre stress. And that R is simply a reduction factor to take away material to make it mechanically not relevant, essentially, once it has been degraded. And this is the result that we got. So we got, for our crude collagenase, we see that our experimental model now explains this linear response that we see in the circumferential direction versus the axial. And when we just degrade collagen, in this case and target it, we get this response. And this is because of this gradient difference in the collagen fibre crimp throughout the wall. And this explains the experimental results that we saw. So using that model explained that, yes, collagen, the underlying collagen degradation mechanism in arteries is the same as that which is seen in bovine pericardium or reconstituted collagen or rat tail tendon. It's just its arrangement in arterial tissue is different, so therefore the arterial tissue degradation is different. So using that, the model is the only way we could really see that. So does this have some physiological relevance? Well, as I mentioned, and I'll show you now, we used dog bone specimens for that to see this tissue, and that exaggerated the crimp difference across the vessel and we did stress relaxation experiments. But in a full intact vessel, the likelihood is at physiological levels, the crimp should be quite uniform across the vessel. And this could change at high pressures to low pressures. And we want to see, does this actually have a difference? How will this affect the degradation of collagen within a tissue? And again, why is this relevant? Because in scenarios like stenting and so on, we expand these vessels really high. Okay, and this could have consequences for degradation of that tissue. And that could be a stimulus for excessive production, which is the case of in-stent restenosis, for example. So it could be, or fibrosis of tissues. So we see here, we carried out inflation experiments. In this case, the way that this was done, so again, for any of you doing experiments, this is a nice setup where basically the vessel itself is cannulated either end. It is controlled by a syringe pump there, which controls the pressure being applied. And this would be either physiological saline for our controls or this collagen, the purified form of the bacterial collagenase. And this is all it has a pressure transducer that can monitor that pressure, and the syringe pump then adapts through this PID control to keep maintaining the pressure. And we put a kind of stochastic pattern onto this vessel and used a digital image correlation system to view the tissue so that we could see the strains within the tissue. So as it is degrading over time. And that's just an example there of one vessel being inflated and our pattern, our strain pattern established from looking at that stochastic pattern on the vessel as it's inflated. So you can see that there. And these are some of the control results. So this is just showing you that as you'd expect, when you pressurize a vessel, it reaches a certain strain and then it kind of levels off if you maintain that pressure at that strain. You can see this creep performance. So here we see that our strain settles off now to a value here over time, and our DIC can establish that strain level. So that's our circumferential strain at the different, over time, for these different pressure conditions that we wanted to investigate. So now we knew we had good, strong controls. We would see if anything would happen under the bacterial collagenase. And here you can see the results for bacterial collagenase. When we inflated those vessels with bacterial collagenase, what we see is that at the high pressures, we have high strains, and as our previous results would suggest, we would expect that the collagen would degrade much more preferentially at these high strains, and we will see failure, essentially, of the tissue because of the lack of presence of collagen here. So you can see at the lower strain levels down here at the lower pressure levels, I should say, which gives rise to lower strain, at 60 millimeters of mercury, we see very much the same as the control. This can be explained because actually collagen fibers are not actually taking the load in this scenario. Very few have been recruited because it's at such a low pressure level. The higher pressure levels, we're getting the collagen fibers being recruited, and that's giving rise to accelerated strain in the collagen fibers at the different pressure levels, and that's why you see this high pressures, these earlier failures. I haven't the result here, but what's interesting to note is, do we see this degradation being prevented in collagen fibers when we're down at the low pressure levels, or at the physiological pressure levels, I should say, and then if we go lower, do we see preferential degradation of collagen? Actually, we do, and the way we confirmed that is we took these vessels here after being exposed to a lower pressure level, and we then exposed them to a higher pressure level, and we looked at the time to failure, and we found that actually they did fail sooner. So even though You can't see it in these curves because they're not pressurized enough. The collagen had degraded, more preferentially again at these lower pressure levels. So looking at this particular outcome that we had got from our arterial tissue specimens, we don't see that here. Now we're seeing that just with increasing strain, we're seeing this increased degradation. So we're not seeing this complex response. And again, why is this? Well, we could use a finite element model, full finite element model, to understand a pressurized vessel. What's, again, the crimp in the fibers within this vessel? It has been suggested that this should be uniform across a vessel at physiological levels. And in fact, if you look at, it makes sense, if you pressurize the vessel, we have collagen fibers within the vessel. At the outer wall, there's kind of the brown, and the inner is the red. They should be in that physiological range. When we remove the load, that changes our crimp. Our outer wall now has, and our inner wall crimp has gone down into this higher degradation. And if we open it out, as we do with our specimens, our dog bone specimens, we get back, this kind of switches, we get back this higher degree of crimp on the outside. And we confirm that with our finite element model, that actually that would happen if we start off at our physiological level, having the uniform crimp throughout. And using this, we were able to now create a full finite element model of a vessel that we could look at, well, what would happen in this vessel if we inflated with different pressure conditions, just like we did in the experiments, and would we see this preferential degradation? And as I will show you here, there we go, we see that under those 4 different pressure conditions, our finite element model, based again on that same model I showed you earlier, that the same premise of this array of fiber matrix units, we do predict this behavior. So we can see that we predict now the behavior of the arterial tissue. What can we do with this? Now we have this model, explains our experiments, we have really good experimental data. We can use it to maybe look at something like an aneurysm to have some clinical application. And this here, we see aneurysms degrade. Typically what you see is elastin will degrade initially, but that will progress to more accelerated collagen degradation. So we wanted to see would our model be able to kind of capture an element of this. So if we had the vessel thin because of initial maybe elastin degradation, would that have a consequence for the collagen degradation subsequently, given using our model? And we know that actually, again, if we allow these to progress, they will end up in eventual rupture. So in this case, it'll move along for me there. Here we go. Here's our model. We just thinned it. We also investigated reducing the stiffness, and it had, again, the same effect. So what we see is that if we have a region of thinned, what will happen is because of that thinning or reduction in material properties, it will have a knock-on effect on the strains that the collagen fibers see, and that will become a runaway process. So essentially, it could explain the kind of growth of an aneurysm through this process of strain-induced collagen degradation. And so this model could now be used to investigate perhaps changes, maybe stiffening up of the collagen, inducing additional collagen, how much collagen do we need to lay down, what kind of interventions should we be carrying out to prevent this runaway process. So that's just showing you again that that is the process. At that proximal region, you'll see this progress towards rupture. And so just to summarize that study, we saw the same collagen behavior at the level of collagen that was seen in other reconstituted collagen, rat tail tendon. We saw a very unique response to arterial tissue, which is all down to how collagen is laid down within the tissue. And we were able to develop a model to explain that. And now we have a model that can actually explain what we see at the full vessel level and can be used to investigate potentially interventions that would go into an aneurysm, for example, in this case. So, sorry, this is going on. So essentially what we're seeing here is the application here is in aneurysms. If we see increased strain, that will have accelerated degradation, will reduce the stiffness further, leading to further increased strain, essentially going on, running away on that increased degradation path, and you eventually could see rupture of the vessel. So that explaining that process. So now this model could be used, I suppose, really to, now that we understand it, to also help in earlier detection and optimization of treatment for these aneurysms. So I'm going to take questions at this stage on that study before I move on to the damage one. I'm not actually finished at all yet. Just as like before, I'm going to kind of move on to another study, but because I think I still have plenty. I still think I have 20 minutes, haven't I, from when we started? I should do. Yeah. So if there's just questions on that degradation before I move on to damage and then into the plaque modelling. Does anybody at this stage just on anything that I've presented so far? Maybe because yeah, sure. No, this is in the model or in the...
In the model.
Yes, sorry. So we were looking at, so we actually measured fibre alignment, yes, but in the model, we're looking at the fibres, yes, being predominantly in the circumferential direction, because having measured them, that is the direction that we would see them in porcine carotid. Yeah.
It means you used the by default alignment. You didn't modify.
No, we didn't look particularly at the fibre angle alignment in that model, in the 1D model. Yeah, exactly. But then in the full FE model, we have fibre alignment there. You can put in the angle. It's a whole Zapfel type model. You can put in the angle of your fibres at any point. So you can change that. So if you wish, yeah.
So have you observed something different? Because in 1D model, you didn't modify the alignment, but in 3D model, like?
No, simply because in the 3D model, we have placed the fibres in the circumferential direction again, because that is what in the porcine carotid in the media, we found that were predominantly in that direction, okay? But if you were to change the fibre angle, you would get different results. So you could do this for different vessels that have different fibre angles.
Sorry, I didn't understand why you put alignment in 3D with the same direction as you put in 1D.
Simply because our measurements indicated that. So if you look at the media data that we got from using our small angle light scattering, that particular artery, as is the case in some arteries, like it's been identified in the iliac as well, is predominantly one family of fibres very much aligned in the circumferential direction. Whereas if you look at other arteries, you do see higher pitch angles, to the circumferential direction, and you see even two families of fibres or, yeah. Okay, so good question, thank you. Yep.
Maybe just also, what's the role of elastin here? Because
yeah,
we haven't been talking about it.
The role of elastin is key for ultimately the strain that this collagen fibres will see, because they're embedded in both gags, and elastin there, is there in networks, okay, arranged so that if not there to take the load, collagen will typically be recruited earlier.
And in your sex data, you don't think elastin plays a role? Or?
In the cells data, we don't look at elastin because you actually can't see, because it's more of a network of fibres, you won't see a clear direction, as in the nature of cells works on a diffraction pattern of aligned fibres. So you won't be able to determine elastin directions from that. But we have done, we'll say, the multiphoton, the second harmonic generation imaging and histology to also look at elastin. But we particularly focused, as I said, and I know, elastin definitely does play a role, and particularly in areas like aneurysms, where its initial degradation is what gives rise to the strains that the collagen sees, which can then give rise to degradation. But we didn't want to focus on that simply because it's not the most critical aspect for load bearing. It's not, for example, in stenting and so on, it's not what will dictate the load-bearing capability of the tissue in the same way in terms of for rupture or, for aneurysms or for, like we will see for the plaques. It's really the collagen content we believe is the key for preventing rupture. Whereas obviously elasticity is key, elastin is key for the elasticity.
And what about the pulsatile load?
Pulsatile load, yeah. We didn't investigate pulsatile here, but again, I would expect you'd see the same effect, as you would increase the pulsatile, if you were to look at pulsatile for strains, look at it at rather than simply look at one static level of strain, but rather to look at the overall physiological strain level. As you'd move up, you should still see further degradation. What you're doing is you're kind of uncrimping and straightening the fibres again, so it depends, I suppose, the range maybe of pressures as to how long it remains crimped or how much it's strained. It's a more complex process, I guess. But again, the same thing, once they're crimped, they're going to be degrading more if they're very crimped. They're going to be degrading more if they're very stretched. So if your pressure levels are very different, maybe that will result in, that should result in really accelerated degradation, hugely accelerated. Do you know what I mean? But again, that would have to be further investigated. But it's a good question.
Because it's interesting when you look at hypertensive patients, some have just very high peaks and others have also high kind of average values.
Yeah.
So depending on the pulse.
Yeah, so our model, this is the key thing with the model. The model could investigate that. Definitely it can investigate how often or how little are you seeing one low strain level versus a higher strain level. And that's the benefit of models, I guess, is that you can test a lot of these hypotheses. So it's a good question. We should try it. Yeah. Okay, thank you. Okay.
There's another question.
Okay. I'll take one more and then I'll move on to the next section because I still have quite a bit. I hope I have time.
We are eating into your 20 minutes.
Yeah.
I have a question regarding the collagenase. So you use probably collagenase B or something from Worthington?
So bacterial collagenase, the crude one is the most commonly used one. I can give you the details afterwards, actually, because we get it from Sigma Aldrich and the exact codes if you want
okay,
it.
yeah,
But,
yeah. So
but the purified one is one that has been used in a couple of studies, and it just happens to be like it makes sure that essentially you don't have any other enzymes present, only the
well,
pure
we use
collagenase.
that as well. So I presume it's a liberase. But do you see batch-to-batch variations?
No, I don't think we have now. Whether we haven't actually gone out and tested. I mean, we ordered it all. I think that particularly the purified one all at one time. We've since ordered new, so maybe we should compare. But
They have a large variation, actually.
Sorry?
They do have a large variation.
You can see. Okay.
We can see. And then I'm wondering, how does this collagenase will eat into through the wall?
Through the vessel wall, yeah. So this is an interesting question, because obviously whether you're perfusing it through under pressure or whether you're leaving it kind of static, this can have an influence, you know, in terms of the diffusion of a collagenase through the tissue. So, you know, that is very hard to account for, and I suppose we could do models to look further at how diffusion of collagenase through the tissue. But what we did is really just do histology over lots of time points and confirm that we had successfully digested away all of the collagen, but not any other components. For both the pure, you know, with the crude we saw, as I said, more than just the collagen being degraded. With the purified collagenase, we confirmed with histology that we didn't see anything else, and that we had sufficient time, so it's like 42 hours, to make sure we had sufficient. And then we did the same experiments on the pressurized one, which takes much shorter time frame, obviously, because we're pushing the fluid through the vessel. But yes, it is. We can't account for the different pressure, at the different pressures, how diffusion will affect the collagenase. Yeah. We just have to make sure we have enough time for all of them to degrade. Yeah, so good question though. Okay, so I'm going to move on to another section, if that's okay. And then again, I'm around, as I said, to answer further questions for anybody. And this is again looking at the idea of in vitro and in silico. And I'll try and be quick on the first section because it's published. But again, to highlight, this is the work of a PhD student, predominantly Dr. Milad Ghassemi, with a postdoc collaborating as well, Dr. David Nolan. So the idea here is really to use a continuum damage model, which again, I won't spend a lot of time on simply because I don't have a lot of time here, but I can talk further on it afterwards, but it is published. But really to use this model in a kind of an unconventional way, and this is to understand how fibers in, for example, plaques here in the carotid, the loading or the stretch that we place on the collagen fibers there can actually give rise to this softening and so on, and that a continuum damage model can actually be used to assess how our fibers are aligned in an optimum direction or not. Okay, so this kind of approach wouldn't have been taken before. Sorry, I should mention again there, just to say, I suppose the reason for this is again regarding this whole area of carotid plaques breaking off, the risk of them rupturing, which gives rise to strokes, which again is one of the leading causes of death from those who die from cardiovascular diseases. And that's an image down there of a carotid endarterectomy procedure, just showing you the carotid as it would appear if you open your neck and you see this carotid, and that's what is cut down and a plaque removed, and those plaques we obtain as well. So to talk briefly about the damage model itself, again, damage modeling was mentioned there earlier, so this model is not, I suppose, unlike that in respect of it captures softening. So this is the idea that when a tissue is loaded and unloaded, it will not go back on the same loading curve. It'll have some permanent softening or change in stiffness. Permanent set, which is this idea of having residual strains. Once I load a tissue again up to a certain level and unload it, you will actually see some, as we would, again, in traditional materials or conventional metals would refer to as plasticity. Here in this tissue, we'll also see some residual strains remain when we've unloaded. And we also see this kind of continuous process of softening that occurs as we cycle tissue, like, for example, in hysteresis, some differences in the load-unload curve. So to capture these in a tissue like arterial tissue, which again has these 3 layers and has collagen fibers, we see this kind of behavior in the overall tissue. This is because of these fibers that are within the tissue. So the fact that, again, they're crimped until a certain critical stage at which they start to be loaded. You see different collagen fibers take up the load at different points, and that's why we see that transition region to, again, a final linear curve where all the collagen fibers are taking the load and eventual rupture of the collagen fibers, okay? So this is what we want to incorporate. Well, is damage occurring in the matrix? Is it occurring in the fibers? I mean, where we see damage in this stress strain curve will indicate what's actually being damaged. But another way of doing it is because we can digest away collagen fibres, we can also actually selectively remove them and see whether the damage is accumulating in the matrix or in the collagen fibres. So that's what we did. Just similar to previously, we took samples of arterial tissue and digested away the collagen selectively so that then we could do mechanical tests on this tissue. So I won't go into that. I've kind of talked a little bit about that. It was all confirmed with histology. We did these mechanical tests, as you can see there, where we loaded the tissue, both when it had collagen fibres degraded from it and when it didn't. And we were able to compare these kind of digested samples, which are mostly elastin and GAG, versus those that had the collagen. And these were cyclically loaded so that we could see if this softening, if this residual strains, this permanent set, and the hysteresis was occurring in this tissue. So this is the digested samples. What you can see is there is some degree of softening occurring, and there is a degree of anisotropy as well within this tissue. And that's an image actually of the elastin structure that's really taking the load in these tissues. The lower plots here are, again, now this is for circumferential and axial. These are intact samples, and these, what look like black down here now, are actually the digested samples that you're seeing kind of blown up here. So we can see, compared to our fully intact samples, the level of softening or damage accumulation in the elastin and GAG, so without any collagen present, is very little. So it's really the collagen that's seeing that softening behavior with those multiple loads. And this is confirmed as well for permanent set. When we compared just the digested samples and the undigested, so the fully intact tissue, and in the circumferential and axial directions, we saw that permanent set was really given rise to, not by collagen fibres, but by the matrix. So that was interesting, that the permanent set is attributable to that. So we developed a model. Again, this is published, but if anybody wants to ask any further questions on it, please come up to me afterwards, rather than maybe going into too much specifics here. This is, again, a fibre-based material. It's a Holzapfel type material where we have a matrix, the collagen fibres, and actually also within the more isotropic, you can see there, if I bring up, you have elastin fibres as well, here represented now by symmetrically arranged fibres. So it essentially is acting isotropically because these are symmetrically arranged. But then we have collagen fibres which have a clear alignment, giving rise to this stiffening of the tissue. We use this model then of with this matrix, these elastin fibres and collagen fibres, to create a damage model where a damage variable evolves over time. And that damage variable is dictated by these parameters gamma and beta, which are for continuous and discontinuous damage. So this idea of damage that accumulates, so we can see here discontinuous and continuous. Damage that essentially, one, that accumulates all the time with every load, even with the unload, and those that accumulate only when we exceed our previous history of our stress, essentially, okay? So just showing you that here, you can see if we imagine the loading, we have to get up to a damage threshold before we'll have any damage. And then when we go beyond that damage threshold, you'll see that your discontinuous damage and your continuous damage over here on the right do increase. Now, when we unload, we don't see any additional damage incorporated into the tissue. But now, as we load back up to where we previously were, we can see that we don't see discontinuous damage, but we do see the continuous damage. Until we exceed that load, will we see discontinuous damage, or gamma increase, okay? So these two parameters are just essentially accumulating damage and essentially seeing, you know, as you accumulate discontinuous or continuous damage, your gamma and beta will evolve to soften the tissue and to induce permanent set into the tissue. So this model, as I said, there's one for the elastin fibres and one for the collagen fibres. So in the digested tissue, we don't have any collagen fibres present. The model is the full model there. The damage model captures just the softening induced in the elastin components. And here you can see that, so the fit to the model from and the experimental data. And then incorporating, looking there just before that to see that permanent set, because as I said, this is part of the matrix, not the collagen fibres, we do see permanent set is induced in the tissue, and it just increases with increasing load. So again, looking at the fit to the full intact tissue now, because we've really a lot of damage accumulating in our, using this damage, the DCF, so the collagen fibres one, and we again can confirm that the permanent set is present, but it's not increasing because of the presence of collagen, because it's mostly in the matrix. So that's just the model itself. So summarizing just that it has permanent set or softening, the permanent set rather than softening is induced at the matrix level. The softening of the arterial, particularly the damage accumulation through softening and hysteresis, is present because of the presence of the collagen. And finally, just that that didn't come up for me, sorry. Oh, it's flicking off, sorry. They're just that this algorithmic gamma and beta can capture the evolution of those variables, can capture the level of damage that's accumulating within the tissue. So what can we do with this? Well, again, we have now understanding around the tissue itself. So in the adventitia, this was work done by Krasny et al. It's very illustrative of kind of the changes in the structure of the tissue. With adventitia, when we apply stretch in one direction, in this case in the circumferential direction, you'll see that you get straightening of fibres in that direction because they're very loosely arranged. Where in the media, you'll again see it aligned that way. But because fibres are loosely arranged in the adventitia, even when you stretch it in the axial direction, those fibres will go into that direction. But that doesn't really happen in the media. Here you can see, okay, because again, they're not as loosely connected. These are really enveloped by gag and elastin and cells. And in the last, they're just showing you again at diagonal. So this reorientation of fibres can occur, and it can be driven by the loading on the tissue. In fact, this is pretty well established. We've had previous damage models as well, which are around the fact that actually physiologically, if you look at the structure of an artery, at even a bifurcation, as I'll show you, the arrangement of the fibres is such that it bears the load in the optimum way. Just like you were talking about optimum, for bone. In the same way, fibres, because of the load-bearing constituent, will arrange themselves to be nearly circumferential, but not totally. And it depends on if they have axial loads on them, that pitch angle, how oriented it is towards the axial direction will be dictated by the axial stretch that's on that vessel. So we implemented this model again, looking at what's the optimum fibre arrangement. Well, it's dictated by the ratio of the intermediate principal stress direction to the principal stress direction. So if we've only stresses in the circumferential direction, our fibres will be pretty much arranged in the circumferential direction, if that's our really dominant principal strain direction or stress direction. And again, as we put an axial load on it, we'll see that pitch angle increase. So that's our optimum fibre direction, and we've an evolution towards that optimum fibre direction. That's our reorientation or algorithm. It will evolve towards that value. So we also have dispersion, which is how variable these fibres are. That also evolves towards the optimum dispersion, which again is dictated by that ratio. So just seeing that evolve to that fibre pattern. So if we look at this, first of all, just in an idealized vessel, applying different axial stretches to the vessels, just 0, 5, and 10%. I'll just quickly show you the interesting bit about this is that we see, as we see physiologically, that the outer wall, so this is just going through the thickness of the vessel, we have a higher angle than at the inner wall. This is relative to the circumferential direction. And we see that if we look at the evolution of what we call the angle here, sorry, just say before that, the angle here, we can see is just over time is evolving to that optimum angle and that optimum dispersion. So what does that mean for the stretch in the fibres? Or what we introduce now, this kind of pseudo-invariant here, this I star, which can evolve just like the damage variables can. So what do we see? We see that strains go down as our fibres align to the optimum direction. That makes sense because now our load-bearing component is moving to take the load, and so our strains will come down. This might result in stresses going up because the tissue has actually stiffened, but the strains going down, this is what matters when it comes down to a plaque, for example. So a carotid plaque sitting here, if it experiences very high strains, which has been seen, this can be linked to vulnerability. Essentially, your plaque is now at greater risk of rupture. So a model like this that can see where the optimum fibre direction is and how much it takes to evolve maybe to that optimum fibre direction from where it actually is physically can be an indication of how at risk a particular plaque might be. So we did that with full vessels, so full final models here of a carotid bifurcation. These were imaged vessels, pre-endarterectomy, the diseased ones. This is actually a healthy vessel. I'm not sure, I don't think this is my one, but it's one of the ones in the group. Here we can see, first of all, we just confirmed that our remodeling metric would predict fibre patterns that are physiological. So here we can see that in this case, if you can read the blue indicates both low dispersion and low angle, which means now we have fibres really highly aligned and close to the circumferential direction. So here we see that most of these fibres then are green here. This is indicating they're near circumferential, but they're not exactly. And that's what we see in a bifurcation. What we see is highly aligned fibres here at the apex of the bifurcation and near circumferential, but at somewhere between, anything from 5 to 10 or 15 degrees, maybe even up to 30 degrees as the circumferential direction in a bifurcated vessel in the straighter sections. So using this, we looked at this for a couple of vessels. we were able to look at these for different axial strains and see that, yes, in fact, our dispersion and our fibre angle will go more towards the axial direction as we increase axial strains. And you can just see this kind of just displayed in this way here. This is 2 vectors representing those families of fibres. And you can see that just as we go from lower to higher axial strains, you can see those little vectors representing the angles of the fibres. And this is quite, again, physiological. This has been seen predominantly, I think by our group who've done it now with diffusion tensor imaging, this has been seen with histology and confocal, that these are the types of fibre patterns, and with multiphoton, that these are the fibre patterns at a bifurcation. So we did this then with a diseased vessel. So again, how is this clinically relevant? Well, in a diseased vessel, we want to say what is the optimum fibre pattern, and then what if those fibre patterns are not optimized? How far are we from that? Can we measure that? Can we put a number on it? Can we understand it? So we developed these models again these were of patients pre-endarterectomy. And so here we can see the fibre patterns that can be predicted from our reorientation or remodeling algorithm, and the dispersion as well. And we do this for multiple vessels, and are able to see it, and particularly around a plaque. You will see here, around that plaque as we move through it, there's quite a complex fibre arrangement needed. So you need to understand this is the fibre arrangement that's needed to take the load optimally to bear the load of the blood pressure within this vessel. And the blood pressure is again obtained for that individual patient. So, well, what can we do? Well, it is known that fibre patterns are different in different plaques. This is a study based on diffusion tensor imaging, which hopefully I will be able to just touch on briefly. And this is to show you that that technique can look at the fibre patterns in a plaque. This is an endarterectomy plaque, and the fibre patterns are different in each one of those, some longitudinal, some more axial. So we looked at 3 case studies, one where we placed all the fibres in the principal fibre direction, 45 degrees to the principal fibre direction, and in the third case at an angle such that it aligned with the intermediate strain direction. And we saw what is the remodelling required to get to the optimum fibre direction. How much will that remodelling be? And this is, again, it's based on a continuum damage model of allowing this beta and gamma to evolve and see how much softening essentially would occur, how much damage would be accumulated to reorientate those fibres. And that's basically the remodelling metric there is simply your D, your damage parameter. And doing that, we were able to see that yes, depending on how far you are from the optimum, as you'd expect, you will see much higher damage. So this has relevance now to essentially be able to say, if we know the fibre pattern in a plaque, and we know what it should be to bear the load, we can see how far one is from the other, and therefore we can have a measure of vulnerability. Now, the only problem with this, I would say, in that idea, is that while we can do this for multiple vessels that are imaged non-destructively, we don't know the fibre pattern of the plaques. Okay, so we don't know these in vivo. So how could we do this? Well, we can see that if we change the fibre patterns before reorientation, we can reduce the strain, so that's key. We can definitely make it less vulnerable by aligning the fibres. So the key thing, whilst all of this can be done, we can get a damage model to determine how much remodelling, give us a remodelling metric. We don't currently have a way to say what the plaque in one particular individual, we can take their geometry and predict what their fibre patterns in that plaque need to be. But until we take out the plaque, we don't know what they actually are. Okay? But this technique now, these future directions, relates to non-invasively determining those. And I mentioned this because I suppose this is the work that we're currently doing, and Brooke, one of my students, is here and she'll be presenting a poster on this, so if you want to go down and have further discussion on that, it would be great. So this is on diffusion tensor imaging and the potential of this to really see things like this structure in vivo. So if you haven't heard of this before, diffusion tensor imaging is an MRI-based technique that looks at diffusion of water molecules in tissue. And you can imagine if you have water diffusing in tissue that's totally isotropic, everything's, it's homogeneous, it's the same, the water will diffuse in every direction in the same way. But if we have a tissue that has structure, so has fibres and cells all in bundles aligned, we should see diffusion of water along those directions, and that should give us an indication of the structure. And this has been done for brain to look at mapping of neurons. It's been looked at in the heart for mappings of the myocardium of the actual cardiac cells, so the cardiomyocytes. And we've applied it to artery as well to see, can we see these bundles in the tissue and the structure? And we apply multiple gradients, so you apply magnetic gradients in multiple directions, and using that then you're able to look at how the water diffuses in a particular direction, and that gives you this underlying structure, okay? We're only doing it ex vivo at the moment, but we believe the key to seeing this, that is this structure of the plaque, critical to the vulnerability and then ultimately translating it in vivo, is this, we first have to establish, is that the key? So we started this in 2010, where we were the first to show the feasibility of just doing diffusion tensor imaging and seeing this underlying structure of artery. We then progressed to show that yes, in fact, if you froze the tissue, for example, which bursts cells and disrupts the structure, could you see it with diffusion tensor imaging? And we confirmed you could. And we have since as well looked at in a bifurcation, for example, using our remodeling algorithm that predicts the fibre pattern. So that's the blue is our simulated remodeling algorithm predicting where should the fibres be to optimally bear the load. And the red is our measured, this is in an intact bifurcation, this is a bovine bifurcation. And again, this has not been done before because all other structural techniques that would have looked at the fibres pattern would have been histology or so on and would have required sectioning. And we see we get a really good match between these two. This is non-destructive. It has the potential to be translated in vivo. I stress that, I suppose, because we haven't done it yet. But even in ex vivo, it has huge potential in understanding the mechanisms. Is it that it is the collagen structure or the arrangement of cells or cell content, for example, that gives rise to the vulnerability of a plaque? And by comparing it to these in silico models, we really can answer those questions. So I'd refer you to Brooke's poster, which is down in the poster session. It's just the first poster board, and we'll be down there to discuss it later as well. Before I finish up for questions, but I first of all thank my group, who obviously all of this work only happens because of them and the funding that I receive. So thank you very much. Probably don't have time for questions, do we?
So thank you very much, Caitríona. We're actually here to listen to speakers and make questions. So if we're a little bit late for lunch,
Okay.
that's fine.
Okay. Hello. Thank you for teaching us so much. I have a couple of very fast questions. So, in your view, what is the level of the tissue strain and the formation of the tissue in a vessel correlated with the flow that passes through the vessel? Does the hemodynamics play any role, or is negligible in your view?
Yeah, I'm always prepared for these type of questions because it's always the case. I've left out the elastin, I leave out the flow, so everybody wants to know why, or does that play a part? And of course it does. I mean, the flow is what dictates the strains that we see in the vessel physiologically. And I think the whole point of this is to understand, I suppose, that when we go outside of those physiological strains, that we are going to get into areas of, pathological mechanisms, because that's where these cells that produce collagen and so on shouldn't even see these strains or have this environment. So the flow plays a key role, absolutely. And we see that, with bifurcations and so on, it's not a coincidence that, again, it's there, the areas we see accumulation. But the mechanics, the solid mechanics also play a role on the effect on flow and pressure. So whilst, if you think about all of these applications, flow will be affected. So if it's mechanical mismatch, compliance mismatch in bypass arteries, that will affect the flow as well as affecting, the compliance and those changes. And I think those both have that exaggerated effect of affecting the mechanical environment that the cells sense and the mechanobiology that goes with that. So we do look a lot at mechanobiology as well. So at actual cells, if we strain them and we incorporate those into models, also at looking at how the cells, when they strain, when they sense pathological strains, how that influences their proliferation, their apoptosis, and so on, and how that has a knock-on effect. But the structure we find is absolutely key. We've done experiments where basically we strain cells on no structure, substrates with no structure, and with structure. And structure always dominates over strain. Okay, when you've no structure, essentially, you know what I mean? So structure is what will always be the dominant force, I think, in terms of keeping cells aligned and determining the mechanical environment that they see. So I think it's always critical for even in the flow environment.
Okay. Another one, faster probably. How long does a diffusion image of a vessel section takes?
Yeah,
because would it be feasible to do it like clinical routine in like 3-4 years, 5?
Yeah, see, this is the thing. I would say long term. I mean, we are finding out there's only our group, as I started this, and there was another group in the Netherlands that did some work in this and have since kind of stopped a certain amount, and then is another group in Austria. So there's not many people doing this in arterial tissue. And one of the barriers would definitely be its movement of tissue and it's the length of time to get the information that you need. So we are talking about for these ex vivo scans, anything from, you can do a short scan in an hour where you'll get a limited amount of information, but actually to get the full gamut of information on, say, for example, a plaque, our scans can run like for 24 hours, you know, anything from 10 to 24 hours. So they really can be very long scans, actually. But using the modelling, I believe we can find what's critical. And again, we may therefore be able to reduce back. So we're doing other imaging techniques in comparison to fusion tensor imaging as well, things like susceptibility and so on. And again, trying to find something that's more translatable maybe in vivo. But it is doing this type of imaging ex vivo that's giving us the understandings that we need to know what's critical for vulnerability, for stability, essentially.
Okay. Yeah, finally, just a note. I do agree with Bart. I think it would be super useful to do the pulsatile pressure, because you can see for how long does it take to damage the tissue in physiological conditions.
Yeah, I agree. And again, this is where I would say models are very useful, because I would say, you know, we'll say what you want to model for is what you can't predict to, you know, kind of easily yourself, you know, to actually work out and go, so, okay, if I have a linear model, I can know what the result is. But a model like this, where we could actually do the experiments, one experiment, and see, does the model predict like that we have collagen fibers crimp for a period of time and then stretch for a period of time. This is where it would be really powerful, where if we could confirm that the model can capture that. I think, yeah. So we need more experiments. Yeah, sure.
How do you manage collagen content?
Yeah.
Because in the experiment, it's quite critical, you know, how much and what collagen density you have where. So how do you manage this also through the modeling?
Yes, yeah. So collagen content, we haven't focused that much on collagen content, to be honest. We're taking vessels from pigs and trying to get, I suppose, the numbers that therefore make sure it's relevant, you know, that there isn't huge variability. But I would agree with you. I mean, collagen content can be key for, you know, the mechanical response as well, for any of these things. So it's probably something, yeah, maybe we need to control for further in the future. I know it's an interesting thing we found in valve tissue, for example, that where we had fibre, we didn't expect to see this, where we had fibre contents very highly dispersed, where we had collagen fibres very highly dispersed, we found that actually those areas were higher in collagen content. And that in itself can actually mean that the tissue can bear load better. We have fibres that can align, and we've enough of them that essentially we can see better performance. So I think, yeah, I probably need to start looking at this more in arteries as well as to collagen content. We've digested them down and looked at collagen content with hydroxyproline and things like that, and imaging. We don't see huge variability on these samples, because our cells, we do histology comparison as well, and we can quantify collagen with that, and we didn't see any major variability that gave us concern. But maybe we should, you know, look at this with in a bit more depth.
Right, because maybe you just use, you know, constant density all over your models now, right?
Yeah,
that's
yeah, exactly. And again, where it could play a key role, yeah, absolutely.
Exactly.
I mean,
What
if
is
there's
the
more collagen to take the load, yeah.
What is the method of choice then that you use to get collagen content?
We've used hydroxyproline, but I have to say it's very slow. And so what we've been using, we did a set of experiments where we compared our hydroxyproline kind of quantitative assays to image analysis and found really good comparison. So from now, we're just always using the image analysis approach. So do histology sections and determine what amount of tissue and then how much using PLM is actually collagen. Yeah.
Okay, last question. Yeah.
Just with regard to the imaging, when we look at fiber structure, we have been looking at aorta mainly and then heart tissue. We have very good experience with X-ray phase contrast imaging using synchrotrons. Did you ever look at that? Because you could do even your kind of dynamic experiments you could do while looking continuously at the structure.
Right. Okay. Well, I'll talk to you about that afterwards, perhaps, and understand. But this is with the synchrotron, so you have to book time at it. Whatever. Would you think you could actually?
But it's interesting enough that I'm pretty sure you can get time. So.
Yeah. Okay. Great. I might chat to you after this then on that. Thank you. It'd be great. Okay.
Okay. So.
Thank you.
We have to move on. So thank you again, Trina.